Hi and welcome. My name is Natalie Caldell and I have a lesson today to talk to you. This is the second of the two lessons on abstraction. The first one talked a little bit about other engineering disciplines and the notions of abstraction for forward and reverse engineering. Today what I'd like to do is apply the abstraction hierarchy that we began last time applied to biology and walk through the different layers of this hierarchy and give some examples. So what we left off with for the first conversation about abstraction is the notion that to design complex systems, it's helpful to break them down into manageable layers so that the complexity can be addressed. And the invention that we have for addressing this complexity is this abstraction hierarchy. Here's one for biology. It may not be the perfect one, but it's one that might work, where at the top layer of the abstraction hierarchy you have systems, complex biological systems, which beneath them are composed of a series of devices. Devices themselves are composed of parts, and parts are encoded by DNA. So that's the top to the bottom view of the abstraction hierarchy. And what I'd like to do is work now from the bottom on up. So with the idea that DNA can encode parts, let's look at what that means. So the best way perhaps to start is to say that a part can be defined as a sequence of DNA that has some human-defined function. So based on the genetics, the biology, the genomics information that we have about that piece of DNA, we can say it does something. The arrow that I'm showing here is designated as the TET-R promoter. That means that there's a protein that can repress this promoter. It's a tetracycline repressor protein, can repress the promoter, but when that protein is not present, the promoter is on. And below that green arrow are a string of G's, A's, T's, and C's. And so the question becomes, how do we know which of those G's, A's, T's, and C's are responsible for encoding that part, the TET-R promoter part? And so, you can ask an expert, you can do genomic sequencing and sequence comparisons between different organisms, you can study biochemistry, and there are answers to which pieces of DNA encode particular functions. And so here I've boxed arbitrarily some of the sequence to say this sequence is what will define the tetracycline resistance protein promoter. Okay, similarly, you might be able to define a ribosome binding site, so that would be a sequence on the DNA, actually on the RNA, that the ribosome would recognize and start translation, but that ribosome binding site is a DNA encoded function or part. Similarly, if you had an open reading frame that encoded the TET repressor protein, that might be what I'm showing here in the pink boxes. And then there could be transcriptional terminator sequences, pieces of DNA, where we know that when RNA polymerase is chugging along, transcription will stop when the RNA polymerase finds those sequences. So in this way, you can define which pieces of DNA are responsible for encoding which of the parts. And as a follow-up to this notion, I'll mention that one of the BioBuilder lab activities, called the iTune device, is a nice way to explore the notion of parts and the consequence of mixing parts. It allows for you to study the output of a combination of promoters and ribosome binding sites. But going back to the abstraction hierarchy, we've moved from DNA to parts, and hopefully you see how sequences of DNA can be used to define parts. And now what we'll do is walk up this abstraction hierarchy to show how assemblies of parts can be used to make devices, and then finally devices can be assembled to make a system. And the example that we'll use for assembling parts into devices and devices into systems comes from the banana smell generating bacteria, which at the system level, you'll remember this is an iGEM project from MIT in 2006. What they wanted to do was make the normal stinky smelling bacteria smell nice during different phases of their growth. During their stationary phase of growth, they wanted a banana smell to be generated, and during the log phase of growth, they wanted a wintergreen smell, a minty smell to be generated. And so the way they accomplished this in terms of devices was to have a banana smell generating device labeled BGD in the second panel down, be controlled with a stationary phase promoter. So when the cells hit stationary phase, that promoter would turn on and it would start to make the banana smell. And what was interesting, one of the many things interesting about their iGEM project was to generate the wintergreen smell. They might have decided to just find a log phase promoter and make the wintergreen smell turn on during the log phase of growth. But instead, they took the existing stationary phase promoter that they had in hand and inserted between the stationary phase promoter and the wintergreen smell generator an object called a genetic inverter. And we'll look inside what are the parts that are used to make the inverter and how does it work. So we're going to be focusing in on this area of the abstraction hierarchy. So with the notion that parts make devices, what they wanted to do was make a device so that when the cells reach stationary phase, the wintergreen smell would turn off. And so that's indicated in this truth table as a 1 or true or on for stationary phase would give a 0 or false or off for wintergreen smell. And similarly, when the cells are not in stationary phase, so that's a 0, the wintergreen smell would turn on. So that would be a 1. And the way they built this was to put actually a normal log phase promoter in front of the wintergreen smell generating device. Now, for those who are botany majors or interested in plants, I will acknowledge that this picture that I'm showing here in this slide is actually of bok choy, not of wintergreen. I don't have a picture on my computer of wintergreen, so I apologize, but bok choy will be our stand-in for the wintergreen smell. And they control this, the transcription of this gene with a promoter that can be repressed by the tetracycline repressor protein. Okay, so that was part 1. Then what they did upstream of that to make this controlled by stationary phase and the absence of stationary phase was they put the stationary phase promoter called OSMY upstream of a ribosome binding site, an open reading frame that encodes the tetracycline repressor protein, and 2 transcriptional terminators. And so hopefully what you can see is that when the stationary phase promoter is on, so when that yellow box called OSMY starts to make transcripts and therefore proteins, it will make the TET repressor protein, which will then bind to the TET repressible promoter upstream of the wintergreen. And that means that when OSMY is on, or gives a signal of 1, the wintergreen smell should be off, or giving a signal of 0, which is what we're showing up in the truth table. And the reverse holds true too. When that OSMY promoter is off, it will not be making the transcript for the TET repressor protein. And without the TET repressor protein, the TET-R promoter that is off on the right side of the screen, driving our bok choy / wintergreen, that will be on. And so the truth table holds true by this very clever device called the transcriptional inverter. And so what we can do is to put a black box around those parts, the ribosome binding site, the repressor protein, open reading frame, the transcriptional terminator, and the repressible promoter. If we put a black box around that, we can call that a genetic inverter, or we could call it a NOT gate. And we can show that when what comes into that inverter gives a signal of 1, the things that come out of the inverter, the signal out of the inverter is 0. And when the input is a 0, the output from that inverter should be a 1. And so now this part, this genetic inverter part, will be useful in any situation where we have a particular input and we want the output to be the opposite. Okay, so that is a way in which parts can be assembled into devices, and I've shown a simple one. There's a collection of these kinds of parts and devices in a couple of places. One of them is the Registry of Standard Biological Parts, which is used for iGEM teams to build genetically encoded machines over the summers or in their high school iGEM teams. There are other repositories for parts. There is one at the Joint Bioenergy Institute in California, where not only do they have a parts registry, but they also have a computer-aided design tool that allows you to choose the correct parts, an automated assembly notion for how you might put the parts together in an automated fashion, and then a redeposition of the parts into the registry. And then finally, there's a biofab facility also out in California, which is responsible for characterizing parts and giving good data so you know which parts to choose for building your systems. And so with this part of the talk, I hope that what you can take away is that you might be able to build biological functions from parts and devices, and that this is a different kind of way to engineer biotechnologies, that when you've wanted to build something before, you've sort of taken pieces here and there and tried to assemble them in an ad hoc way. But by building with repositories of parts, it's a lot more like building with electronics or things like that, where you have characterized elements that you can assemble to generate particular functions. So that is a quick walkthrough of the abstraction hierarchy, where we've gone, in this case, mentioned how DNA can be defined into parts and how parts can be assembled into devices. And just like the abstraction hierarchy that we talked about when they built the Boeing 777, all the same elements are true for this engineering concept, that everyone has to be doing their job right for the final product to work. If I build a genetic inverter that doesn't work very well and you use it to build a system, your system won't work well. As the engineered complex systems for biology get more and more complex, the banana smell generator is reasonably simple. You can imagine building something super complicated. There may be no single person that understands everything about that final product. And in that case, actually, the abstraction is really important for knowing where to study and probe and optimize the system. And then finally, as with the 777, the interfaces between these layers of the abstraction hierarchy are really important. There's not a reason to build a device that no system needs or that no parts exist to build it. So there has to be good conversation between the different layers of the abstraction hierarchy if, as an engineering system, it's going to work. So this is one of the tools that was suggested early on in the BioBuilder lessons to be important for making biology easier to engineer. So abstraction is the black box idea of defining inputs and outputs and having elements that you can then build with without having to understand all the complexity and all of the details. One way to apply this abstraction hierarchy and to see its usefulness is to start to develop systems of your own. So if you start with a system that you are particularly interested in, be it a system to address food and energy needs or health and medicine, foundational advances, all sorts of things are possible. It can be an interesting exercise to walk through the abstraction hierarchy from your system into devices, from devices into parts, and from parts to DNA elements. So we can do that in another lesson, but hopefully this quick lesson on the abstraction hierarchy and how you black box particular elements in order to manage their complexity and be able to use them in many settings has been clear and has been interesting. And thank you for listening, and I hope to see you soon. Bye.